P-500 expression levels are exceeding the baseline projection.
Cross-reference the metabolite synthesis against the historical data, just to be sure.
The precursor levels have stabilized ahead of schedule.
The P-500 cultures are ready. Reroute to industrial processing immediately.
The P-500 cell cultures are transferred from the seed bioreactors. This aseptic process is inside the intermediate bioreactor suite, an array of 100-liter and 500-liter stainless steel bioreactors.
All parameters verified. Sterility confirmed.
Excellent. Initiate full-scale production sequence. Agitated by powerful impellers, the dense cell culture synthesizes synthetic venom Model P-500. Cell viability remains high. The neurotoxin and hemotoxin graphs are spiking.
Confirmed. The targeted substrate delivery is working.
The entire contents of the Synthetic Venom Model P-500 bioreactor is carefully pumped massive pipes.
The analysis confirms the structural integrity of the P-500 Synthetic Venom's neurotoxin components after lyophilization.
This meticulous labeling ensures full traceability from raw DNA to the purified powder, a crucial aspect of reorganization guidelines.
The liquid synthetic venom Model P-500 undergoes final sterile filtration. This precision ensures absolute sterility.
The newly sterile filtered liquid synthetic venom Model P-500 flows through hermetically sealed conduits towards the aseptic filling machine's reservoir.
This data point confirms the efficacy benchmarks are met, ensuring viability for drug development.
The receptor binding data looks consistent across all plates.
Yes, both the neurotoxin and hemotoxin components are verified.
The data is conclusive. It meets all regulatory standards.
Agreed. Final approval granted.
Seals are secured on Container 7.
Temperature reading is stable at -50. Ready for transport.
A vast logistical network ensures prompt and secure delivery, connecting advanced manufacturing to the global scientific community.
Ensure thermal lock is engaged on all P-500 containers.
Confirmed. Bay 7 secure.
A precisely measured dose of Venom Matrix injectable is introduced into the cell culture. As the venom
We're observing clear signs of neurotoxicity. The viability metrics are dropping faster than expected at this concentration.
receives. Robotic systems screen thousands of antibody fragments against the synthetic neurotoxin and hemotoxin components.
The P-500 matrix shows strong interaction
Action with the TLR4 pathway.
but the neutralizing antibodies had limited efficacy.
The Venom Matrix shows significant neurotoxic effects crucial for antivenom development.
Have you analyzed the hemotoxic pathway interactions yet?
This synthetic venom model, P-500, is the breakthrough we needed.
Absolutely. The acceleration of drug development is unprecedented.
The Venom Matrix is showing high efficacy in the latest trials.
And the robotic analysis confirms the structural integrity of the P-500.